Well, good morning or afternoon, depending on your time zone. This is Carl Pilcher, and I'd like to welcome you to the new season of NAI Director Seminars, and it is really a pleasure to kick off this season of seminars with a seminar from Norm Pace. Norm, as I'm sure many of you know, most of you know, is an extremely distinguished microbiologist. He is a member of the National Academy of Sciences, and in 2001 he received the Selman A. Waxman Award in Microbiology, which is given by the Academy and is generally considered the nation's highest award in microbiology. That same year he also got a MacArthur Fellowship just for good measure, and just this year he received the American Society for Microbiology's Abbott ASM Lifetime Achievement Award for outstanding contributions to the field of microbial ecology. I have to express personal appreciation to Norm because over the years that he has been involved in exobiology and astrobiology, which is quite a few years, he has helped me a great deal just in understanding microbiology since I wasn't educated in it. And so it's with really great pleasure that I introduce Norm Pace, who's going to talk with us about life in the universe, the expanding world of microbial diversity. Norm, take it away.
Norm, can you unmute your microphone?
Let's try that again. The title is slightly changed from that Carl gave you. I changed it to Life in the Universe, the terrestrial example, because I believe that we are one example of a lot of life out there in the universe. Life in this planet has penetrated every possible thermodynamic and physical niche, and the requirements for life anywhere in the universe are going to be the same as they are here. So I think we're seeing a way that life accommodates to life in the universe. Now, the history of life on Earth, of course, began about 5 billion years ago when a star out on the edge of a galaxy, not too different from the one shown here, except that our own 5 billion years ago exploded in a supernova, scattering star guts over perhaps a cubic light year of space. We know that we're a second or third generation star because of all of the heavy atoms that are in our solar system, including carbon. The heavy atom carbon only formed in the hearts of exploding stars. So we expect life anywhere in the universe to be based on carbon. And those who might like further discussion of that, I refer you to the National Research Council report recently issued on the limits of organic life in planetary systems. And I think there's general agreement that life chemistry will be organic chemistry. So in talking about life's diversity then, I first have to give you a metric of how to think about diversity, a molecular map of life's diversity and evolution. And we'll then take that perspective and use it to go into the natural microbial world to find out what kinds of organisms are on this planet. It turns out we haven't known very much about that because microbiologists have always had to culture organisms, petri dishes and all that. And it turns out we can't culture much of what's out there in the real world for many reasons. But by using this new perspective, a molecular perspective, we can go into this natural microbial world and explore that which we haven't known very much about, which I tongue-in-cheek call dark life of terrestrial biology. Using this technology and perspective, I'll talk about expanding this tree and give you a general perspective of life's diversity that certainly hasn't hit the textbooks. And then finally, I need to give you a public service message, which will become clear later. Our textbooks aren't doing it very well these days in terms of biological diversity. Well, biological diversity, how to define it? You've heard a lot of talk about biological diversity over the years, but did you ever hear anybody define it? I haven't in a satisfactory way. However, recently it's become possible by the use of gene sequences to use sequence comparisons to study phylogenetic trees, relatedness genetically, relatedness trees as metaphors for evolutionary diversification, to make maps of evolution. People have been thinking about relationships between organisms for a very long time, of course. This is a phylogenetic tree, if you will, from Ernst Haeckel in 1866, only a few years after Darwin. And you'll see it's pretty much what you see in textbooks today. Down here at the bottom we have Monera, and then animals and plants, and protists, unicellular eukaryotes. In the 1960s, fungi were plugged onto this to give the familiar Five Kingdoms tree, and the name Monera was changed to Procaryote. Nothing else happened. I'll return to that. Now, this is a subjective view of life based on the kinds of organisms that Haeckel could understand at that time, a subjective view of life. And it really wasn't until we had a molecular view, only about the past 30 years or so, has it been possible to have an objective view of life's diversity. And this is through the techniques of molecular phylogeny. It's about relating organisms by gene sequences. And this is a simple process. In principle, you get the sequences that you can compare from different organisms that you want to compare. You very carefully line up those gene sequences and count the number of differences. And that number of differences will be some measure of evolutionary distance. It's then an issue based on those pairwise distances to calculate some sort of a map, a phylogenetic tree that most accurately represents all of the pairwise sequences. Now, you can do this process with any gene that you would like, but if you want to compare all of life's diversity, there are only a few genes that are appropriate. And the genes that have emerged as the gold standard, if you will, for deep phylogeny are the genes for the ribosomal RNAs, highly conserved and present in all organisms. Now, the first person to use ribosomal RNA genes to cast phylogenetic trees of all of life was Carl Woese, then and now at the University of Illinois. And I'll offer paeans to NASA at this time, because NASA kept Carl Woese alive when all the other scientists in NSF and NIH and so forth said, "Carl, this is just taxonomy." NASA knew better, and out of that came an absolutely spectacular picture, a map of all of life, this being one example. So this is a phylogenetic tree which shows examples of all of life. Let me show you how to read it. Here's the scale. 0.1 changes per site, and evolutionary distance is read along line segments. Here we humans are out here. All of animals would fit into this radiation. All of fungi into this radiation. All of plants in the line indicated by Zia. And then, of course, there's the rest of biological diversity, which is fundamentally microbial in nature. Now, you can't find out where the origin of this big tree is from ribosomal RNA sequences, but there are other phylogenetic methods that one can employ, also with biochemical correlates, that show the root is out here deep on the bacterial line, and that means, as cartooned over here, the origin is out here, the bacterial line, and the eukarya and archaea are related to the exclusion of bacteria. And I'll return to that one as well later. Now, just to get a few points on that I want to make sure that I cover. So just a few lessons from the big tree, this map, if you will, this cartoon map of all of life's diversity. There are three main relatedness groups, three kinds of organisms at the deep level: bacteria, eukarya, and archaea. The origin, as I already mentioned, is out here on the bacterial line, or the line leading to bacteria, which means that archaeas and eucs are related to the exclusion of bacteria. And indeed, much biochemistry would say that the many fundamental aspects of the biochemistry of these organisms is far more similar than either is to bacteria. Now, analyses such as these show very clearly that the chloroplasts and the mitochondria are of bacterial origin. Endosymbiotic origins for chloroplast and mitochondria is now very well established. However, the eukaryotic nuclear line of descent is as old as the archaeal line of descent and derived from something more rudimentary than either bacteria or archaea. That means that the prokaryote-eukaryote model for biological organization is shot throughout our books, our textbooks, our literature, and our language. It means that that model for biological organization was wrong. I'll return to that. Most important for the current presentation is that sequences, ribosomal RNA gene or other sequences, are identifiers of organisms, barcodes if you like, for the specific organism. You don't need to culture in order to identify. That's important because microbiology has always been paralyzed. As already mentioned, microbiologists always had to culture organisms to detect and identify them. And you don't culture very much of what's out there. If I go down behind our building to Boulder Creek and pick up a little bit of water and count directly the number of organisms in that water and then do what I can do to culture them, I'm going to culture perhaps 1 in 10,000 of what I see in the microscope. More importantly, you don't culture 99.999% of what's out there, and that's what our understanding of biological diversity has been based on until quite recently. But using sequence as identifiers, we can open up the whole panoply of molecular biology to explore the natural microbial world. So you go out and get yourself a natural sample, you prepare DNA from that, and then use, for example, PCR to grow up ribosomal RNA genes of all representatives in the community. It's always a complex mixture, so you have to clone these ribosomal RNA genes in order to separate them into their individual components, which are then sequenced. You can do phylogenetic analysis if you like, but this is more than a game in sequence collection because you can do something with the sequences. For example, use the sequences that you determine to make hybridization probes or PCR primers to explore various aspects of the natural samples, or chase the critter into culture if that's an essential thing to do. So this opens up a very easy experiment to begin to explore the natural microbial world. You get yourself a sample, you make yourself some DNA, you do some PCR, you clone the products, and you determine sequence. Very simple experiment. An absolutely wonderful experiment because it's easy to do, and you do the same thing again and again and again, and you always get an interesting answer. And so I'm going to give you some examples of some of these interesting examples. First, I want to tell you a little story about Yellowstone because this illustrates a real problem we're up against in studying the natural microbial world using molecular technology. So it's easy to go out and get sequences and do phylogenetic analysis and say, aha, look at all these interesting organisms. But the real challenge now is what are they doing out there? We can tell you who's out there. We can identify organisms in the environment, but how to find out what they're doing. And so this is just to indicate the lump we're up against at this stage. From phylotype to phenotype, from what an organism is phylogenetically to what that organism is doing. How to do that. There's only one principle that you have in studying all of these otherwise unknown organisms, and this is the representatives of a particular phylogenetic group are expected to have the properties that are common to that group. So if we can relate organisms in the environment to those that are known with particular properties, we can lay those same properties on that organism that we don't know anything about, at least in principle. This is an aerial view of Grand Prismatic Springs, large hot spring. It's about 88 degrees centigrade. There's the boardwalk over there with people walking along. It's the largest hot spring of its type anywhere. You can see it's kind of discolored around the edges of the pool, and the reason it's discolored is because of microbial biofabrics that penetrate the center around the sides of the pool. We've done a lot of work in Yellowstone over the years. We don't work in Grand Prismatic because there are going to be tourists walking by, and the Park Service doesn't like us to do that, so we work in backcountry pools such as Octopus Spring here, absolutely remarkable place. 88 to 90 degrees water wells up and flows out. Can't see it in this photograph, but again, it's kind of pinkish around the edges of the pool. If you hang a string across the outflow channel over there, just over the course of a couple of days, this gelatinous filamentous material accumulates. Is it alive? Well, if you take a pinch of that stuff and make a microscope slide which is stained DNA with diamidinophenylindole and look at it in the fluorescent microscope, you see, aha, these filaments bind DAPI. They must contain DNA. They must be alive. And if you look at them in the scanning electron microscope also, you see that this is characteristic of life at 90 degrees centigrade. That is really hot. You do not stick your toe in that. Or nearby Obsidian Pool, where we've done a lot of work. Absolutely remarkable place. Looks like a real origin of life place. It's a hot pool, depending on where you stick the temperature probe, 75 to 95 degrees centigrade, sparged from below by gases that make a real roiling pot kicking up obsidian sand around the side. Real hellhole looking place. But if you take a clean microscope slide and stick it in that sludge just over the course of a few days, the microscope loads up with biofilm. So here after 2 days, 4 days, 6 days, that is very rapid growth on that microscope slide for organisms in the natural microbial world. What are they doing for a living out there anyway? What are they eating? It takes energy to do that. Biology is in the business of capturing energy and using that energy to make more of itself. Where are they getting energy? They're certainly not doing photosynthesis, which doesn't happen above about 70 degrees centigrade or so. Well, as you walk around in Yellowstone, or if you pick up books that talk about Yellowstone, for example, you're usually led to believe that there's a lot of hydrogen sulfide around, as there is. You walk around Yellowstone, lots of steam, lots of sulfidic smelling odor. And oftentimes the textbooks will tell you, if they tell you anything at all about the nutrition of organisms at high temperature, is that, well, they're probably doing some aspect of hydrogen sulfide metabolism. Well, I know that's not the case in Octopus Spring, for example, because I know there's no hydrogen sulfide in Octopus Spring water. So what are these organisms in the hot springs doing for a living? We've been messing around with that question for about the last 20 years, and we've looked at ribosomal RNA and other gene sequences in a lot of Yellowstone hot springs over the years. And when we first started accumulating these sequences, all we could say is that, geez, these organisms are really different from anything that we know about. But then also over the years, cultivars, cultured organisms came out that will now allow us to reflect the kinds of organisms that we see in the Yellowstone hot springs to those which have been examined in culture. And the bottom line is that roughly 80% of the kinds of organisms that we've seen over the years in Yellowstone hot springs are the kinds of organisms that only are known to eat hydrogen for a living. Hydrogen. Who would have thought of hydrogen in Yellowstone hot springs? Well, we've now gone back and measured a lot of hydrogen in a lot of Yellowstone hot springs, and indeed all of them have hydrogen from 15 nanomolar to up to 300 nanomolar in the waters of these hot springs. And even 15 nanomolar is fat city if you can eat hydrogen for a living. So that's one arena we've studied. Another arena that's been of particular interest to the NAI is studies of the Guerrero Negro hypersaline microbial mat. Folks at NAI Ames have for many years been studying this site as an example of an ecosystem, microbial ecosystem, that you could hope to get a take on what's supporting. Guerrero Negro is a small town about halfway down the western coast of Baja California, here seen here in an aerial view. And the interesting thing about it is that there's a salt works there producing industrial grade salt. There's a cartoon, this lagoon, water is taken out and then it evaporates until it moves up through a series of pools to ultimately crystallize. And there's a lot of life in these pools. There's a quite dense microbial mat from 5 to 8 or so centimeters thick, about the consistency of tofu, a photosynthetic driven community, which we've been studying. So here's an example of this tofu. No one's had a nerve yet to have a barbecue with this stuff. In any case, we take core samples and chop these down into layers to study the microbial makeup by virtue of the ribosomal RNA distribution. We've studied quite a few of these now. We have about 140,000 ribosomal RNA gene sequences from this environment, and it's by far the most complex environment that's ever been studied in this way. Here's an interesting community. As you get up toward the high salinity levels, you begin to see salt crystals dropping out along with gypsum, giving a gypsum halite structures like these. It looks like a pretty lifeless place, but if you take one of these chunks and cut it over, then you see that in reality that's shot through with microbial biomass. So this is a chunk of this gypsum halite. So you see the colors, the photosynthetic colors of the microbes stratifying in the salt crystals, and including some sort of larvae in here munching on the microbial biomass. There are lots of eukaryotes in this environment as well, mostly nematodes as it turns out. So we can get a take on the diversity by looking at the ribosomal RNA gene sequence population, just as an example of that. The detail of this isn't important. This refers to the relationship of the ribosomal RNA sequences and the number of operational taxonomic units. So if you summed over this line, you would have a representation of the diversity of the Guerrero Negro microbial mat. And if you summed under this line, you would get the complexity of the human gut or the mouse distal gut, both of which would be considered complex systems, but overwhelmed by the situation in Guerrero Negro. We probably understand why that extensive diversity, because in the anoxic environments, it's represented by these microbial mats. There's no oxygen when you get below a millimeter or two. In these anoxic environments, you can accumulate a lot of biochemical gradients that would be rapidly shorted out, if you will, chemically by exposure to oxygen. And so that high level of complexity of organic chemistry makes for high levels of complexity of microbial species as well. We've done this same business of ribosomal RNA and other gene sequence analysis in a lot of areas over the world. We've studied hydrothermal vents ranging from black smokers to tube worms, for example. We've studied acid mine drainage. This photograph was taken inside Iron Mountain in California. There's water running out of there. It's very acidic, pH 0.1, and loaded with really nasty metals. And yet look at all that luxurious microbial growth being sampled here by Brett Goebel. The open ocean. You think of life in the open ocean, you tend to think of fish and seals and whales and stuff like that. Man, forget it. In terms of overall contribution of biomass in the open oceans, the game is the microbial world. This is a wonderful picture, which I got from Jed Furman. This is a fluorescence pic of a little bit of seawater. It was stained with the dye cyber green, which again binds to DNA. So this is life in the ocean. This is most of it, way more than 95% of life in the ocean. You can see a little bit of chlorophyll fluorescence here in these rather largish, actually, eukaryotic organisms, probably eukaryotic organisms. And you see these little blobs here in this photograph. Those are bacteria, so-called marine picoplankton, each about only half a micron in diameter. And the thing that fascinates me about this photograph is beyond the little blobs of the bacteria are all these little dots, viruses. Wherever you look, the virus to cell count is 10 to 1, wherever you look. And that virus community, of course, is carrying around the metagenome, if you will, of the largest biome on the planet. The ocean is the largest biome on the planet, 70% of the Earth's surface, on average 4 kilometers deep. That compares with our 30% of our aerobic or our air terrestrial, if you will, 30% of the world's surface, only call it 20 meters deep. Largest biome on the planet. Very little understood, very little known about it. It's nice to go out in nature and look at the life. Most people, when they go to Yosemite, would look at the trees and the bears or whatever else is at Yosemite that's big and walks around. I like to look at things like the stains up on the rocks. Stains, pigments from microorganisms living on the surface of the rock. And you'll note it gets black down here by Bridal Veil Falls. That's water drives life. Pigments, photosynthetic pigments. Now, life is out there not only on the surface of the rock. If you took a chunk of this rock and broke it off, actually we won't do it there, we'll do it somewhere else. We'll go up behind my house in the Flatirons, study of graduate student Jeff Walker. Jeff Walker, when he was a graduate student, study of these endolithic microbial communities, endolithic, inside rock. So if you look at a piece of Flatiron sandstone, for example, that's what it looks like on the surface, and if you cut off a chunk, that's what it looks like. Here's the surface, and green stuff and other colors into the penetrating the rock. There's a scale, 1 millimeter, and you can see that the coloration that you can see with your eye extends about a centimeter into the rock. Rock is actually remarkably transparent, or at least a lot of it is. In this photograph, Jeff put a flash up here and then shot it off and took a photograph at right angles to it. And you can see the light penetrates the rock pretty well. In fact, if the rock were wet, so that one could get reflectance around the silica grains, it would go very much deeper. Important point is there's plenty of energy out there. In fact, living in the rock is a good place to live. Some places on the planet, for example, in the dry valleys of Antarctica, that's the only kind of life that you find. Now, these are all rather exotic places compared to us, and so I thought I would show you another project that we're currently involved with, is studying the microbiology of the indoor environment. It's all around us. Here's postdoc Laura Baumgartner collecting air in the New York City subway, which turns out to be a pretty interesting place biologically. Or even this room. If I take a deep breath, I probably suck in something like a couple thousand bacteria, something like that. What are they? We mostly don't know what kinds of organisms are all around us. Or if you happen to take a shower in the morning, whoops, what happened? Oh, man. Well, sorry, guys. That's what you get for using a different computer. So if you take a shower in the morning, and if you look at your shower curtain, you say, gee, there's some white speckles down at the bottom of this shower curtain. We call it soap scum, familiarly. Well, if you scrape off a little bit of that soap scum and put it onto a microscope slide and stain it with a DNA binding dye, DAPE, There's your soap scum. And when you turn your shower on and it blasts up a little bit of that soap scum into an aerosol, you breathe it. What kinds of organisms are those? Well, we mostly don't know. We can actually tell you. We've studied shower curtain soap scums. I can tell you a lot about it. There's a lot of variation. Nothing dangerous unless you're immune compromised or have an open wound. We indeed are embedded in a microbial world, ladies and gentlemen. Well, these studies over the years now really expanded pretty dramatically our view of microbial diversity. So this is just a plot which Dan Frank did. It stopped around mid-2005. Here's the accumulation of sequences of cultured organisms, actually mostly the same organism, lots of E. coli, lots of salmonellas. Here are the environmental sequences now being picking up as the technology has improved. And so in mid-2005, the number of environmental sequences in the database passed the number of those sequences from cultured organisms, and it's really screamed up now. There's probably in the order of 400,000 ribosomal RNA gene sequences in the databases. And this has really expanded our view of diversity as well. What do I mean by that? So this is what's happened to bacterial diversity over the past 20 years. This is a cartoon of bacterial diversity. Each of these lines itself is a radiation of different kinds of bacteria. These would represent the main file of bacteria, the divisions of bacteria, different kinds of bacteria. It's a cartoon of diversity, if you will. When Carl Woese could first do this summary in 1987, at that time there were 12 of these divisions, all of which had cultured representation, none with uncultured representation. After a decade, things really began to expand. We were up to about 36 of these divisions, about 20 of which are cultured. They're indicated by the bold lines. The lighter lines are environmental sequences that have no cultured representation. By today, 2007, we're up to about 100 of these bacterial divisions. Only about 30, which I think is generous, have cultured representation, and 70 or so have no cultured representation at this time. And of these 100 or so bacterial main phyla, only 7 have any significant cultured representation. And those are the clades, those are the relatedness groups that contain a pathogen. Microbiology study of diversity has heavily been directed by medical relevance. So this is a cartoon then of what I think the big tree looks like at this time. So the bacteria, about more than 70, about 100 at this stage. Just to put some names on these, I won't pick through it. Archaea, 2 main groups seemingly at this time, Procaryote and Eucaryote. And rather more complex picture of eukaryotes and more controversial tree. I put some fuzz around the base of these radiations just to indicate the uncertainty of the radiations. I don't want to get tied up with that at this stage of the game here. So our view of biological diversity at this stage is approximately this with respect to overall genome evolution, if you will. And let's do remember that genomes are more complex than ribosomal RNA genes alone. But this does represent a rough map of the course of terrestrial evolution. Absolutely wonderful. And it's a measurement. It's not a guess. It's a measurement. Rough, to be sure, but it's a measurement. That brings me to my final topic. We need to change the way that we think about terrestrial biological diversity, and it's not being done well at this time. We need to do away with this notion of prokaryote. There isn't any such thing. And it's more than terminology. This is much more than terminology. It speaks to the very foundations on which our biological thought rests, and I'll defend that. I don't care what the scientific discipline is. There are basically 2 things, 2 issues that the discipline worries about. The discipline has to worry about order. And this would be the organization and relationships of the things that you would study. So, for example, the chemist would study the periodic table. The biologist would study phylogenetic relationships. The other fundamental thing that any science worries about is how what one studies changes, how things evolve. So, for example, the astronomers would study the Hertzsprung-Russell star series to understand the evolution of stars, the function of stars. The biochemist would study biochemical mechanisms. The chemist would study radioactive decay. And biologists now have a metric with which to understand biological change, the tree of life. There's the tree of life. I don't see any prokaryote on there. I don't see anything about prokaryotes on there. That's a measurement, a map, not a hypothesis. A rough map. So an important issue of that map is that the prokaryote-eukaryote concept, two kinds of organisms, prokaryotes giving rise to eukaryotes, is wrong. Flat out wrong. Flat out wrong idea for the essence of biological thought, organization, and evolution. Well, here's where it came from. I already mentioned that to you. This is where prokaryote, eukaryote came from, right out of the 19th century. So Haeckel had his Monera down here, plants and protists and animals. As I already mentioned, in the 1960s, a mycologist by the name of Whittaker plugged on fungi because he thought they were important, and indeed they are. And the name Monera was changed to prokaryote. Nothing else happened. Prokaryote means non-eukaryote, nothing more. Not being something is not a scientifically valid concept or name. There ain't no such thing as a prokaryote. No one can tell me what is a prokaryote, only what it is not. Well, we're scientists. Let's test that prokaryote-eukaryote model. And here's what I mean by the prokaryote-eukaryote model. I think that most students coming out of university education this is what they believe. Here's a cartoon. The cartoon is that prokaryotes are a group, and they gave rise to eukaryotes. So all eukaryotes are of a kind, i.e. all eukaryotes would fit into that triangle. All prokaryotes are of a kind related to each other to the exclusion of eukaryotes. That's prokaryotes. There are two forms of cellular organization and function, prokaryotic type and eukaryotic type. Prokaryotes is kind of a subline here. Prokaryotes are simpler than eukaryotes. And finally, that prokaryotes gave rise to more advanced eukaryotes. And there's any number of models where archaea fused with bacteria and who knows what. But prokaryotes gave rise to eukaryotes would be the model. We now have a database with which to test that model. It's put onto the table again, essentially in the 19th century. Modify. Whoa. So we can now test this notion of prokaryote, eukaryote, and here's the model that we can test it against, or rather the data, the map that we can test it against. So all eukaryotes are of a kind, specifically related to one another. That's true. All euks would fit into that triangle. All prokaryotes are of a kind, related to the exclusion of eukaryotes. That's false. There are two kinds of them. There would be prokaryotes out there, one of which is more closely related to eukaryotes than either of those is to bacteria. So that's false. There are two forms of cellular organization, namely having nuclear membranes and not having nuclear membranes. Well, there may be two forms, there may be three forms, there may be 16 forms for all I know. I do know that cellular organization is more complex, far more complex than simply presence or absence of a nuclear membrane. I think you shouldn't poison the question by edict that there are two forms of cellular organization and function. At least the scientists shouldn't. Prokaryotes are simpler than eukaryotes. Well, I argue that's not really an evolutionary issue at all. It has to do with niche occupancy. Yes, it has evolutionary flavor to it, but if I take a 2,000 gene Prochlorococcus and a 30,000 gene dog and drop them both out into the largest biome on the planet, the open ocean, which one do you think is going to make it? It's not going to be the dog. And finally, the concept that prokaryotes gave rise to eukaryotes. That's false. The eukaryotic nuclear line of descent is as old and derived from something more rudimentary than what became archaea. So the eukaryotic nuclear line of descent is not derived from either archaea or bacteria, rather from something far more rudimentary, and certainly from neither archaea or bacteria. Why am I incensed at this? Some of you out there will have heard me give you something like this story before. Why am I incensed about this? Well, it's because truth is important, and prokaryote is scientifically unjustified. It was invented to fill a gap in knowledge. We didn't know what that little stuff was, and it was all dumped into Monera, a.k.a. prokaryote. The name has false implication in deep evolutionary matters. The textbooks flat out will tell you that prokaryotes gave rise to eukaryotes. Baby. The prokaryotes, if you will, the bacteria, gave rise to mitochondria, no question, and gave rise to the chloroplast, no question, but not the nuclear line of descent. So this false understanding quenches legitimate inquiry. It is interesting to know what means cellular organization in archaea, because it probably speaks to our own rather than bacteria. And here's the one that really bums me. Prokaryote eukaryote teaches our students false concepts at the most fundamental levels of biological order and evolution. Biological order and evolution should be the foundation for biological thought and for progress in biological thought. And prokaryote eukaryote muddles the issue. Well, what else to call them? Well, that, of course, depends on what you mean by them. If you mean the little stuff out there, try microbes, microbial. I've stopped using the word. It's hard to stop using that word prokaryote because it's so ingrained. But try microbial, microbes. And there's a lot of little, weird, mostly ignored eukaryotes out there, too. No sense in dissing them when dealing with the microbial world. If you're talking functional or evolutionary issues, you need to be more precise than prokaryotes. So, for example, how many textbook chapters are there on prokaryotic RNA synthesis? A lot of them. And they're not very meaningful because they speak only to bacterial RNA synthesis or bacterial protein synthesis. For example, whereas bacteria use sigma factors for transcription initiation, for promoter selection, so bacteria use sigma factors, and we, eukaryotes, we don't do that. We use so-called Tata binding proteins, a different mechanism. So do archaea use Tata binding proteins. And crystal structures of the archaeal Tata binding protein inform our structures of the eukaryotic version. So prokaryotic RNA synthesis, prokaryotic protein synthesis, that's an oxymoron. It doesn't mean anything. Humans conceptualize in language. Our students conceptualize in language. We order our thought in language. Thus, in the language is the understanding. Prokaryote has to go. It's intellectually not defensible at this time. Those who would defend prokaryote simply don't understand deep issues in biological evolution. Thank you, ladies and gentlemen. A lot of people have been involved in this project over the years, and I list them here, not only the studies that I told you, but a lot of others as well. So thanks to you all, and I'll be very happy to answer questions. Floor to Carl.
Norm, thank you very much. I would just like to ask everybody who just heard this great talk to remind colleagues who weren't able to hear it that it will be archived on the NAI website, and that should be up within a day or two. And I'd like to take advantage of having the open mic to ask the first question, and that is, Norm, what conclusions do you draw from everything you've told us about life off the Earth?
Well, Carl, I don't think we can say anything about life off the Earth except by the extrapolation that we expect life anywhere in the universe to be organic chemistry. It's likely that there will be minor differences from the kinds of life that we see on Earth, but I bet not a lot, and I would love to see, by the way. Again, the life anywhere has the same constraints as life on Earth, namely to grab energy and to convert something, CO2 ultimately, into organic material. Life on this planet does this in a number of ways, and so I think if we see not the detail, then at least the generality of how life would accomplish this any place in the universe. Beyond that, it would be babble.
Thanks, Norm, and I think I'm going to turn it over to Marco. If you have a question, would you please raise your hand on WebEx, and Marco will call on people with raised hands. Do we have some raised hands?
Not right now.
Well, since we don't have any raised hands on WebEx, would anybody like to jump in with a question?
Okay, it's Mike Mooma at Goddard. Boy, Norm, that was a fantastic talk, first of all. But beyond that, I really liked your analogy of the dog in the ocean. And what it really made me suddenly jump back in my chair and think about was, okay, maybe in fact the highest life forms have evolved to fill such niches that are so specific that if you take them out of those niches or modify the niche in some way, you then threaten the very survival of the life form itself. And of course, we're not going to throw 6 billion humans into the ocean, but what if to what degree do you see that we might actually be modifying those other ecoforms that permit us to occupy this unique niche that we have?
I think that that's really an excellent point from two standpoints. Number one is we humans are wrecking the planet, and I sometimes to my students like to refer to the lash of Gaia, which we will one day feel, and really already are. The other thing the microbial world offers is an analogy in the sense that there are basically two kinds of organisms. There are organisms that conduct primary productivity, that is, capture some sort of energy and convert CO2 into biomass. The second kinds of organisms are so-called heterotrophs. These are the organisms that use the reduced carbon to drive themselves. We humans need to take note of that as we continue to dig up and modify the planet in order to get it reduced carbon that we can burn in our automobiles and in our power plants. We humans need to begin to note that, as with biological settings anywhere, we have to be able to harvest exogenous energy. And for us humans, that means light or nuclear energy, and we have to move to that as soon as we very possibly can, because, to return to the first point there, we're rapidly poisoning the planet, either by CO2, by other organic output from human activities. And again, we humans need to pay attention or we won't live very long. End of comment on that.
Well, yeah, I hit several key points, but another one that I think is really extremely relevant is the issue of what unforeseen consequences might you as a microbiologist think of in terms of genetically modified plants whose products we eat. And one wonders whether, in fact, over time, this couldn't cause internal genetic modifications that could, in fact, in a relatively blink of an eye, change the ability of humans to actually survive.
Well, I think we humans are pretty tough creatures unless we do it ourselves. I think, yes, things like genetically modified plants could introduce genes into the environment. At this stage of the game, at least in terms of U.S. federal regulations, you're not supposed to introduce modified genomes into the environment, but of course it happens all the time. And of course modification of genomes in the environment happens all the time. Need one mention only the spread of antibiotic resistance on Earth's surface. Our last hopes for antibiotics are rapidly going away, and we certainly need to explore new ones. So just in summary, I think we humans have indeed already had a major impact on the planet. We've forced extinctions. And, I used to be more optimistic about humankind getting their act together, but now I'm less optimistic. I rather fear that over the next couple of 100 years we're going to turn the planet into what is modern-day Haiti. And need one only mention the ongoing population problem, which is not even part of the political discourse, it seems to me. So to repeat, one could go on about this, but I think that, yes, we have a lot of evidence for human impact. Yes, we can see a lot of problems with regard to altering the biosphere. It will not necessarily be of good consequence to humans.
Norm, Nick Wolf in Arizona. As you have you've got gene sequences from more and more organisms. Presumably you're starting to expand those 3 triangles that you have. What would you expect to be a sign that you have found the place where archaea and eukaryotes meet?
Well, archaea and eukaryotes meet all the time. Your guts are loaded with them, for example. We have a lot of methanogens in us. Those are archaea. So if you're looking to see what the distribution around the planet might be, from the many environments that we've looked at now, if I go out and grab a handful and do a ribosomal RNA sequence analysis, 90 to 99% of the ribosomal RNA genes I see will be bacterial, 1 to 10% will be archaeal, and 1% or less will be eukaryotic, unless you grab a nematode, in which you get a whole lot of eukaryotic ribosomal RNA genes. So, and we're all living together all of the time. I don't see that there's any place to look, any need really to look to see where they meet. The most extreme environments, some of the most extreme environments we've looked at, for example, the Guerrero Negro microbial mat, hypersaline mat, or even the salt crystals, there are eukaryotic ribosomal RNA genes in there. Not a lot of them, but that's typical of what we see around the planet. I'm not sure whether I'm answering this question.
I'm not sure that you are either. So let me go back. You had, for example, Giardia sitting at near the tip of the eukaryote sequence, and presumably there could be other things with even fewer inserts into them from bacterial lines. And I'm just wondering what you would be expecting to find as you found things that were nearer and nearer to the origin of the eukaryotes.
Well, we've certainly been looking. We've seen gene sequences of other branches deep in the tree, and they're useful in the sense that it allows us to deal phylogenetically with the eukaryotic tree better. But the phylogenetic tree of the eukaryotes is more controversial, because if you use other genes, you get different sorts of things. The best information at this stage of the game is that such lineage as the diplomonads, which would be Giardia, or the trichomonads, which would include Trichomonas, and some of these deeply divergent lines, that in phylogenetic calculations are not long lines. And so from the ribosomal perspective, there is this basal radiation of eukaryotes, just like you see a basal radiation in the bacteria and a basal radiation in the archaea. Rose has written about this. It all boils down to these long blank lines at the base of the individual domains. Those long blank lines were probably a period of evolution before the invention of sufficient biochemical complexity that the particular lineage could carry an independent line of descent. So Carl Woese has painted those early days of biochemical evolution as being very communal, when genes could move around between replicating foci. However, as the biochemistry grew more sophisticated, this means that macromolecules would be working with other macromolecules in very specific ways, and so as evolution went on, any particular macromolecule could less easily be transferred out of its own expression and replication focus. And so only when the sophistication was complete, only when the development of complexity was sufficiently sophisticated that organisms could have an independent line of descent, only then could the diversification be seen, and that would be seen as the radiations at the base of the domains. Carl Woese has called that the Darwinian boundary, or Darwinian border, when Darwinian evolution could then weigh. The question also oftentimes comes up is, well, geez, are you going to see a fourth or a fifth domain? And when we first started this work a long time ago, I was after fourth domains and fifth domains and so forth. But we've accumulated a lot of ribosomal RNA gene sequences from a lot of places, and we've seen lots of archaea and lots of bacteria and lots of eukaryotes, and we've never seen anything that even smells like a fourth line of descent. I don't think it's out there at this stage, or if it is out there, we can't detect it with our tools, but I'll quickly be the first to admit that the sorts of tools we use, namely ribosomal RNA or other molecular-based tools, are all based on organisms of the kinds that we have already seen. So are there other lines out there? I think it's a possibility, but at this stage I don't think it's likely.
Thank you.
We have a question from University of Washington.
Yeah, I appreciate the talk. I think you made it accessible, a very sophisticated presentation, accessible to simple country geologists, and I really appreciate that. A question that comes to me, however, is where on this planet would you go to find the most primitive life form possible? Or what possible exploration strategies would you put in motion now for getting there 5, 10, 15 years from now?
I think that there's no such thing as a primitive organism alive on Earth today. All forms of life alive today are the products of 4 billion years of evolution, all exquisitely fitted to the niche that they occupy, and if they didn't, somebody would eat them. So I don't think that there's a primitive organism to be found anyplace. Simple organisms, yes, simple. Now, it turns out the simplest free-living organisms that we know of at this stage of the game are found in the open ocean. These are organisms such as the cyanobacterium, Prochlorococcus, and other kinds of organisms that live out there in the open ocean, and they're doing it on 1,500 to 2,000 genes. Or methanogens, for example, in anoxic environments are making a living off of eating hydrogen and breathing CO2. Can't get much more rudimentary than that. And again, some methanogen genomes are in the order of 1,500 to 2,000 genes. So I think that those are the simplest, or the kinds of organisms that one can find any place, and probably the fewest number of genes that one can operate on anywhere in the universe. But primitive, they're not.
Well, because the environment has been stable for 4 billion years?
Well, no, it hasn't. It's changed, and the life has changed along with it. Now, I would have argued not too long ago that, yes, if we go, it's probable that life came to be in geothermal environments, and so if we went into geothermal environments, we would find organisms that are particularly primitive. And the fact is that you don't find primitive organisms. And by primitive organisms, I mean those that would either branch very deeply or have very short line segments, and therefore be most closely related, statistically at least, to the common ancestor. You do see short line segments in some types of organisms that would occupy extreme environments. For example, the Procaryote that live at high temperatures are not particularly simple genomes. They typically will have 4,000 or 5,000 genes. And they do, however, have relatively short branch lengths in trees based on ribosomal RNA. I think, however, this short branches does not reflect primitivity or less evolution. I think it represents more limited sequence space available to high-temperature organisms than to low-temperature organisms, namely constraints on structure enforced by the temperature, not by evolution.
Thank you.
I have a question that is there any analysis done based on not the phylogenetic classification but the system of bioenergetics, like how minimum energy that a life can utilize and survive and proliferate, something like that?
Well, bioenergetics is pretty common throughout life's diversity on Earth. Organisms use ATP metabolisms, organisms use chemiosmotic energy harvesting. All kinds of organisms do that. The types of energy sources that are being used, of course, hydrogen, hydrogen sulfide, all kinds of reduced compounds, those are found in many, many kinds of organisms. They're not particularly phylogenetically limited. Organisms harvest light in different ways. The Calvin cycle, of which everybody's probably familiar, ribulose bisphosphate carboxylase and all that, that is indeed an important mode of photosynthesis that you see distributed in a lot of kinds of organisms, but it's not the only way to do it. The reverse TCA cycle, in fact, may be more common, certainly as common in the microbial world as is the Calvin cycle. We know very little about the distribution of that. Proteorhodopsin's another big game. It was thought that that rhodopsin had its origins in archaea, since it was first recognized there, but it subsequently turned out that rhodopsins are present in a lot of bacteria as well. First discovered by Ed DeLong in the context of proteorhodopsin, but now seen as a lot of places as an alternative way to harvest light. Not as trick as chlorophyll-based photosynthesis, but it works. So I don't know what to say about that. I don't see particular phylogenetic constraints on the kinds of energy energy harvesting and energy utilizing mechanisms that are employed by terrestrial life. And again, anywhere in the universe, I would expect to find not exactly the same solutions, but similar solutions.
But is there can you see any trend if you classify based on bioenergetics, how much energy they use or something like that, then don't you see some trend besides phylogenetic trend? I mean, like a tree of life, the evolutional trend.
Yeah, I don't see any particular trend. Similar kinds of energy metabolism are used by a lot of different kinds of organisms. If you're trying to get at the question of what's the best energy source, the best energy source is probably the most rudimentary, thermodynamically speaking. Probably the best way to make a living in terms of harvesting energy from the simplest stuff is to consume hydrogen and breathe oxygen. Those are at the extremes of the thermodynamic ladder, and probably the most efficient way to make a living. And a lot of organisms do that.
Thank you.
We have another question here at NAI Central.
Hi, Norm. This is David Morrison. I want you to help me with a class of questions I get to ask an astrobiologist from what I presume are high school teachers who are bothered by whether the kingdom concept still means anything in terms of modern domains, whether there are kingdoms, whether that applies to any of the organisms outside the eukarya. Can you help me with that?
Well, it's an issue of historical nomenclature, David. And indeed, when the original 4 kingdoms of life were defined, or 5 kingdoms of life, that was what was known. And since then, we've learned that the tree of life is way bigger than that subjective view of life's diversity. So kingdoms was originally a name which was given to what were considered to be the major provinces of life. We now know that there are a lot more of those provinces, but even the definition of a kingdom is a little bit messy. There are about 30 phylogenetic, what we would call clades, relatedness groups. About 30 big relatedness groups in the eukaryotes, for example. Are those all kingdoms? Well, I would say so. But most professional systematists, most professional taxonomists are still in denial about what's happened in terms of the explosion of our view of life's diversity, just frankly in denial, and hasn't been dealt with. And, well, the bacterial, Rose, when he first wrote about bacterial diversity, described the bacterial divisions or phyla, whatever you want to call them. He used also the metaphor of kingdoms to describe those of the bacteria, or the metaphor of kingdoms to describe the 2 major kinds of archaea that we currently recognize. So there isn't a straightforward answer to your question, David, because it's so muddied by history. For a professional systemist to agree that there is a kingdom of diplomonads, you would probably find to be a professional systemist would say, well, that's not really one of the kingdoms that we historically recognize. Hey, I agree with that. But on the other hand, there's as much, at least ribosomal RNA diversity in the trichomonads or the diplomonads as there is among the animals, all of the animals. Now, what means biological diversity? We use these ribosomal RNA trees as a metaphor for diversity. And I'm quick to add a metaphor for diversity because biological diversity is far more the biological diversity of the organisms is far more than let's call it relatively simple pictures painted by the phylogenetic trees of ribosomal RNA. But I do believe that the ribosomal RNA tree gives us a backbone of large-scale evolution of terrestrial life.
Okay, does anybody else have any questions that they'd like to pose to Norm? Looks like we have one Goddard.
Yeah, I'm Ginter Koltechka at Goddard. You showed this beautiful picture of the ocean water, which showed these organisms. And you mentioned, oh, this might be prokaryote, this might be eukaryote, and there was also these little tiny things that you said that might be viruses, virus DNA. And I was wondering at that time, I mean, how much it is possible that it could be just free-floating DNA, some DNA which just is remnants of some organism that decayed. And if you have any idea how much time would it take for this DNA to decay?
A couple of answers. One, those little specks of light are indeed viruses. They've been concentrated and looked at in the electron microscope, and a couple of labs around the world are studying the metagenomics, if you will, of the marine virus community. And so like I say, you can see them. They're viruses, not specks of DNA. The question of how long free DNA would live in the marine environment is an interesting one, and people have done measurements where they'll take radioactive labeled DNA and mix it with raw sewage or with other kinds of stream water, pond water, ocean water, stuff like that. And the answer is that if you throw labeled DNA into raw sewage sludge, it lasts for about 30 minutes, something like that. If you put it into marine water, it lasts for maybe a week, half-life of about a week. So it goes away relatively fast at the cosmic scale, but the DNA that's within viruses is probably pretty well protected, since that's how those viruses make their living. And like I say, you can draw these down under the right kinds of filters and see viral morphotypes without any problem. It's pretty well documented that those little specks of light represent viruses. I don't think you would see little pieces of DNA. Perhaps a little piece of DNA stuck on a piece of fish scuzz or something, but not that very large number of little spots of light.
Thank you.
We have a question from Blair Hedges.
Yeah, can you hear me?
Yeah.
Not very well. Get closer.
Yeah, hi. Yes, Blair Hedges. I was curious what's your take on the symbiotic origin of eukaryotes, and specifically how that ties into your anti-prokaryote argument.
Would you repeat that? I couldn't hear that question very well at all.
Yeah, many people in the field of early eukaryote origins believe that eukaryotes arose, at least both cytologically and also genetically, through symbiosis. So it's not really a vertical inheritance in that sense. So they would argue that perhaps there is a prokaryote-eukaryote distinction. How do you argue against that?
I don't see any evidence for it. I don't see any evidence for creating a relatedness group of prokaryotes. I do see evidence that there are 3 lines of descent. There's hard evidence that the eukaryotic nuclear line of descent it came out of something even more rudimentary than archaea, and developed as ultimately into what became the eukaryotic cell. Now the eukaryotic cell, of course, took in bacterial symbionts, chloroplasts, mitochondria. That seems to be in the general theme of eukaryotic complexity. Basically, eukaryotic metabolism, energy metabolism, for example, is really rudimentary. It's basically fermentative. The eukaryotes have achieved nutritional diversification by, again, taking in a partner, a symbiont, a bacterium or a chloroplast. When did that happen? When did the earliest eukaryote arise? I don't know. When did the earliest archaeon arise? Well, the eukaryotic nuclear line of descent was in place at that time. Now, that earliest eukaryotic cell did not have mitochondria and chloroplast, because it could only bring those in when the organisms that gave rise to chloroplasts, cyanobacteria, or alternatively proteobacteria, which gave rise to mitochondria. We know that the chloroplastic line of descent came to be as early as the cyanobacterial radiation occurred. That is to say, if I make a cyanobacterial phylogenetic tree which includes chloroplastic sequences, I will see that the chloroplast roots into the base of the cyanobacterial radiation. That is to say, the chloroplast is not derived from any modern kind of cyanobacterium, but rather originated at the same time as the cyanobacterial radiation as a whole. And of course, the chloroplast-less eukaryotic cells were well in place already by that time. Let me go further and say, don't confuse the modern kind of eukaryotic cell that most of us think about, those that have mitochondria and or chloroplasts. A lot of eukaryotes don't have mitochondria and chloroplasts. These would include the diplomonads, the trichomonads, these basal divergences from the eukaryotic tree. Some have argued that these things used to have a mitochondria and then lost them, but the data for that are not good at all. The main reason to believe in prokaryote, eukaryote at this time is because Ernst Haeckel told us that Monera gave rise to eukaryotes. I repeat, in the 1970s, fungi were added to give us the 5 kingdoms, and the name Monera was changed to prokaryote. Nothing else happened, and that's what our children are being taught in high schools and colleges, and I think it's a travesty. Enough said.
Well, I think we'll let Norm have the last word on that, and let's thank Norm for a great talk.